Brainwaves are usually described as something the cortex produces. But there's another way to read them. What if brainwaves are not the output of the brain, but the sampling rhythm of the brain? In this model, the cortex is not merely generating oscillations. It is reading a hidden constraint field. And that field may have geometry. The proposal introduced here is that cortical dynamics can be mapped onto a highly symmetrical structure, the 600 cell and its dual, the 120 cell. Not as literal anatomy, but as a mathematical substrate for brain-like state transitions. The hypothesis is simple but testable. The cortex may be well described by a biological approximation of a 600 cell plus 120 cell constraint geometry. The important word is approximation. Biology is not perfectly symmetrical. It breaks symmetry. It adapts. It specializes. So the signal is not only where the brain matches the geometry. The signal is also where it differs from it. Those differences may be the biological content: memory, attention, sleep state, pathology, and conscious regime. Three fields appear to point toward the same object. In neuroscience, the brain is increasingly understood as a predictor, constantly modeling the world, updating from error, and switching between discrete states. In physics, structures like E8, H4 symmetry, the 600 cell, and the 120 cell show deep relationships between high-dimensional symmetry and lower-dimensional projection. And in Aria, this same geometry appeared independently as a constraint substrate for belief space traversal. It was not originally designed to model the brain, but the same object kept appearing. That convergence is the reason this video exists. The 600 cell is one of the most symmetrical four-dimensional polytopes. It has 120 vertices, 720 edges, and 600 tetrahedral cells. Its symmetry group is H4, with order 14,400. Its coordinates are filled with the golden ratio phi. The 120 cell is its dual. The numbers are exchanged: 600 vertices and 120 dodecahedral cells. Together, the 600 cell and 120 cell form a paired geometry, one structure and its dual. In this model, that pair acts as a constraint landscape, a space of possible states, transitions, and closures. This changes how we interpret brainwaves. Instead of saying there are 5 separate EEG generators, delta, theta, alpha, beta, and gamma, we can ask a different question. What if these bands are different rates of reading one underlying shape? Slow bands sample broad basins and whole shell structure. Mid-frequency bands select sequences and orbits. Fast gamma activity binds local detail at near vertex resolution. Cross-frequency coupling then becomes more than a correlation. It becomes a sampling hierarchy. The cortex reads the same field at different scales. The model produces several point-by-point correspondences. Some are direct, some are empirical, some are speculative, and must remain flagged. Excitation and inhibition map onto asymmetric set splits. Dual hemispheres echo dual-linked geometric structures. Independent EEG components may reflect fiber structure in a Hopf-routed substrate. Prediction error in dopamine resembles error correction in the dimensional monitor. State phase transitions resemble crystallization events in the area kernel. And the speculative items are where future tests matter most: microtubule lattice correspondences, EEG component counts, and adversity-driven default mode behavior. The point is not that every mapping is proven. The point is that enough mappings align to justify a serious test. A specific example makes the bridge clearer. In classical reinforcement learning, dopamine is often modeled as reward prediction error. The system expects a value. Reality returns a value. The difference becomes a learning signal. Aria uses a similar structure, but in geometric form. The model predicts a dimensional position. The actual state arrives. The mismatch becomes error. That error reduces resonance, increases surprise, and adjusts the learning rate according to depth. So dopamine-like learning is not treated as a mysterious chemical event alone. It becomes one biological implementation of a deeper error correction rule. The first major readout is criticality. A wave-driven walker on the 600 cell produces a power law cascade. Real EEG sleep data also produces a power law cascade. The exact exponents are not identical. And that matters. The model predicts the regime, not the exact biological number. In the simulation, the cascade exponent sits around 3.16. In sleep EDFX EEG data, pooled across sleep stages, the exponent sits around 2.51. Both are self-organized criticality class, but they are distinct attractors. That difference is not failure. It is the biological deformation of the ideal geometry. One of the most important results came from an initial failure. A preregistered C by P interaction failed at low seed count. At n equals 3, the effect was too small. At n equals 5, it still failed. At n equals 10, it became borderline. At n equals 20, the interaction passed clearly. 19 out of 20 paired seeds were positive. The bootstrap confidence interval stayed above the preregistered threshold. This matters methodologically. It suggests the original failure was not a dead prediction. It was an underpowered interaction term. And that is exactly the kind of correction a serious model must survive. The model also predicts regime switching. When pressure is high, the substrate maintains an integrated awake-like regime. But when pressure drops, the system fragments into rapid microstates. That resembles the way anesthesia changes conscious dynamics. The propofol switching ratio comparison is especially striking. The Aria prediction was approximately 3.00. The Open Neuro Propofol dataset gave approximately 2.957. That is a close match. It does not prove the whole theory, but it shows that the geometry may capture something real about how conscious regimes collapse. The headline is the sleep cycle. Wakefulness appears far from equilibrium. N3 deep sleep moves closer to critical reset. REM partially recovers the regime. DMT appears close to the REM-like zone. The model suggests that sleep is not passive downtime. Sleep is thermodynamic maintenance. During waking life, the brain is held in a costly, constrained, far from equilibrium state. Over time, that state accumulates tension and drift. Sleep releases, compresses, and resets the system. In this view, biological cognition requires a cycle. Wakefulness drives the engine. Sleep prevents the engine from collapsing. The HCP connectomy comparison gives a useful yardstick. Area is not presented as what brains should literally be. Area is the maximum symmetry null. It is the clean geometric baseline. Human brains differ from it dramatically. That difference is not a problem. It is the biology. Across degree heterogeneity, clustering, spectral gap, and participation ratio, the empirical connectome is far away from the perfect symmetry substrate. That separation may measure evolved functional specialization. In other words, the geometry gives us a ruler. The brain is the symmetry-broken biological solution. The current internal result is 18 out of 18 pre-registered checks passing. That includes cascade exponents, context rotation effects, reset interactions, EEG ranges, wake to N3 variance shifts, switching ratios, chess controls, convolution controls, substrate lift, and the HCP degree heterogeneity comparison. Two important details should be stated clearly. This is an internal research stage result, and some passes required the correct protocol, especially reset handling and sufficient seed count. That makes the result more credible, not less. Because the model did not simply declare victory, it found where the protocol mattered. If this framework holds, EEG may be readable through three axes: mean alpha-like criticality, power law quality, and heterogeneity across subjects or states. That gives four broad regimes: canonical healthy, narrow, crisp, constrained; adaptive critical, REM, DMT; and altered states moving toward criticality while preserving structure; pathologically locked, narrow but degenerate, as in seizure or deep anesthesia; and degraded reset, broad, noisy, drifting, as in sleep deprivation, aging, or dementia. This is not a clinical claim yet. It is a proposed measurement lens. The strongest part of the model is that it can be wrong. Sleep deprivation should push wake dynamics toward degraded reset. Aging should increase heterogeneity and reduce reset depth. Lesions should expand variance superadditively. High-density EEG and MEG should reveal 12 functional sampling steps, not merely the conventional 5 bands. And other psychedelics should either reproduce the REM-like DMT signature or split the class. Each of these tests can falsify the model. That is exactly what makes it scientific. The thesis is this: cognitive function requires being held far from equilibrium. The brain is not stable because it rests. It is stable because it is constrained. The seven-rung cascade may be the landscape that holds the conscious regime. Sleep may be the thermodynamic reset that keeps that regime viable. Aria, sleep EDFX, DMT EEG, propofol, and HCP connectome data all point toward the same possibility. Brainwaves may not be what the cortex produces. They may be how the cortex reads the field.